The Scepter Cell Counter is a handheld automated device for counting cells, monitoring cell diameter and volume, and checking the health and quality of cellular populations. It offers significant advantages over other counting methods, including improved accuracy, ease of use, and low cost. To count cells using the Scepter, follow the instructions on its graphical display to aspirate cells into the disposable sensor. The cells are then counted, and the device reports cell concentrations and statistics, which can be uploaded to a computer and analyzed.
Hi, I'm Sónia. I'm a research scientist here at Millipore, and today I'm going to show you how to use the Scepter Cell Counter.
Counting cells is a necessary but tedious step for in vitro cell culture. Consistent cell concentrations ensure experimental reproducibility and accuracy. Cell counts are important for monitoring cell health and proliferation rate, assessing immortalization or transformation, seeding cells for subsequent experiments, transfection or infection, and preparing for cell-based assays. Despite the need for speed and accuracy in cell counting, 71% of 400 researchers surveyed count cells using a hemocytometer. While hemocytometry is inexpensive, it is laborious and subject to user bias and misuse, which results in inaccurate counts. Sources of error in hemocytometry include uneven cell distribution in the sample, too many or too few cells in the sample, subjective decisions as to whether a given cell falls within the defined counting area, contamination of the hemocytometer, user-to-user variation, and variation of hemocytometer filling rate. To alleviate the tedium associated with manual counting, 29% of researchers count cells using